Okay, now I'm going to try and pick up the lecture pretty much from where I stopped off and fill in the missing parts that I was not able to complete. Now I want to go back a little bit and just remind you about a couple of relevant points concerning neutralizing antibodies. These are the antibodies that can prevent interactions between the viral attachment protein, and these are the cell receptor or co-receptor that is necessary for virus attachment to the host cell. Other types of neutralizing antibodies can prevent infection of susceptible cells because they'll fix to the fusion domains of virus attachment proteins, and prevent them from catalyzing fusion of the virus envelope with the cell membrane. So this will be relevant to envelope viruses. Now, because neutralizing antibodies, either of the IgA or the IgG class, can be secreted at mucosal sites. For example, they'll be present in your saliva or at the genital mucosa. This means that viruses that arrive at those sites can be bound by the antibodies immediately, even before they can infect a single cell in your body. That's why neutralizing antibodies, when they're present, can confer sterilizing immunity and prevent infection from occurring at all. Now, it's important to realize that not all antiviral antibodies are neutralizing. So that means a person can be seropositive for antibodies by ELISA or Western blot, but they may not necessarily have developed neutralizing antibodies against the particular virus. This is a problem we're going to look at a little bit later with respect to influenza and HIV. Now, we saw in the lecture a couple of examples of how hyperimmune immunoglobulins or monoclonal antibodies could be used in post-exposure prophylaxis for viral infections like rabies or Ebola virus infection. And this kind of strategy can only work if the same neutralizing antibody will have an antiviral effect against all of the different strains or isolates of the virus that exist. Now, that's not always the case, because some viruses have different neutralizing serotypes. So what does that mean exactly? Well, if you were to do this experimentally, imagine you've got an immune serum from a person who has been infected by poliovirus. And then you prepare serial dilutions of this immune serum in 2 different plates. And then to one plate, you add the virus that comes from this person. Let's call it poliovirus 1. And to the other plate, you add a constant amount in all the wells of poliovirus 2. So when you have the person's own immune serum against their own virus, at some point, you're at high concentrations of serum, and the virus will be neutralized and there will not be a cytopathic effect in these wells. But as the serum gets more and more dilute, then neutralization will not occur and you'll have infected cells. However, if you look at what happens with the virus 2, then no matter what the concentration of the serum is, all the wells still get infected. So that means that poliovirus 2 here is not getting neutralized by a serum which is active against poliovirus 1. And that means that polioviruses 1 and 2 are of different neutralizing serotypes. And this is important because this explains why some acute infections you get once in your life. Smallpox, when it existed, people caught it once and then they were immune for the rest of their lives. The same is true for measles, mumps, chickenpox, and varicella. Whereas for other acute infections, you can get infected pretty much every year. So that's true for adenovirus and rhinovirus, which can cause colds and respiratory disease. If you look at the number of serotype, neutralizing serotypes that these viruses have. The ones where you just have a punctual once-in-a-lifetime infection, you either have one neutralizing serotype or a limited number. So poliovirus has 3 neutralizing serotypes. All of these other guys have got 1 neutralizing serotype. Whereas for these recurrent infections, there are 47 known adenovirus serotypes and maybe more than 100 rhinovirus neutralizing serotypes. And that means that when you get infected with an adenovirus, like B serotype 5, well, you can still be reinfected by serotype 10, serotype 11, and so on. That's where you can get multiple recurrent infections by these viruses. The situation with influenza is a little bit more complicated, so I've kind of left this out for now. Now, another thing that all these viruses have in common, which is not the case for these ones, is that we have an effective vaccine available for all of these viruses. And it's going to work against this year's virus and next year's virus, and in 10 years' time, these vaccines will still be effective against their respective viruses. And that's not the case for what we have that's not the case for the influenza vaccine. And going back to what are influenza serotypes? Really, they're kind of more like serogroups, okay? So imagine you've got a panel of virus strains that have been isolated from different people or at different times. And then you've got a bunch of sera that come from people who have all been infected with influenza. So you know that there's going to be some influenza-specific antibodies present in these sera. So the first serum, and you're going to test, which viruses can be neutralized by these different sera. So the first serum might neutralize viruses A and B. Second serum might neutralize viruses B, C, and D. And the third serum might neutralize viruses D, E, and F. And then these other sera down here will neutralize either, some of these G, H, I, J, and K viruses. So none of these sera. So what you can see is that for the first 3 sera, okay, these blue ones, there's some overlap in their neutralizing spectrum, but none of them can neutralize all the viruses from A to F. And none of these sera neutralize any of the viruses that are down in this G, H, I, J, K group. So all these viruses in group A to F, they seem to have some antigenic similarity. And the same can be true for the viruses from G to K. Okay? So you can define 2 different serogroups on the basis of this kind of neutralizing test. And through other experiments, you can detect that this neutralization will be dependent on binding of the antibodies to the hemagglutinin protein. And then you'll be able to call these different viruses as you'll be able to determine that these different viruses are part of the same neutralizing group. And that's what their hemagglutinin serotype will be. So all of these viruses, A to F, will be part of the H1 serotype, and these G to K viruses will be part of the H2 serotype. But that doesn't mean that if you have antibodies to one particular virus in the H1 serotype, that your antibody will be able to neutralize all of the different viruses in the H1 group. Because in fact, even though they have some antigenic similarity, they are not identical. Now, the fact that the antigenic identity of influenza viruses change over time is the phenomenon of antigenic drift. And this can be shown experimentally. This is an experiment published in 2001. And what the authors did was they took a bunch of different viruses isolated at different times. So this one's from 1968, 1972, 1975, all the way to 1997. And they infected ferrets with them. Then they recovered sera from the ferrets after they had had the infection and recovered, and tested the ability of the serum to neutralize the different viruses. That's where they are down here. So if the ferret was infected with a virus from 1968, it got a neutralizing antibody against the virus from that year, but it couldn't neutralize any of the subsequent viruses. And the same thing went on. Virus from 1972, you get certain neutralization against that virus, some slightly weaker neutralization against the previous virus, but nothing against all the future viruses. And so it went on. So every year, each individual virus can induce neutralizing antibodies against itself, the influenza virus in that particular year. But if you get neutralizing antibodies against that virus, it doesn't protect you against viruses that are isolated in the future, and it doesn't protect you against viruses that were present in the past either. So year on year, the antigenicity of influenza viruses changes. And the motor for this change is continued escape from the neutralizing antibodies that are present in the population that is getting infected. Now, the upshot of this is that the influenza vaccine must be changed regularly. So every year there is a meeting of experts in at the World Health Organization to determine what virus strains are going to be put in the vaccine. And these people have to make their best prediction about what is going to be the dominant neutralizing serotype in next year's influenza season. Okay, so from time to time, every 2 or 3 years, the nature of the influenza strains that are used to put into the vaccine is modified. Okay, so from 1970 to 2002, these were the different strains of H3N2 virus that were used in the vaccine. H1N1 was mutated rather more slowly. That is until we had the new H1N1 strain that appeared in 2009. Anyway, to get back to so this is the reason why the influenza vaccine needs to be changed through time. So as a clinician, you have to remember that you need to get your influenza vaccine renewed every year, because the vaccine you got last year might not be protective against next year's influenza, the influenza virus that's circulating next year. And it's also one of the main reasons why the influenza vaccines that we currently have available are not 100% effective. Influenza vaccine is an effective vaccine, but only when the circulating strain is a good antigenic match for the virus that was used to prepare the vaccine. And if you get infected with a virus that has mutated, and even though the vaccine works, it won't protect you from that new influenza strain. So this is one of the reasons why people may say or may have the impression that the influenza vaccine is not effective. They might come to you and say, "Doctor, I got myself vaccinated for influenza in the summer, but now look at me, I've still got the flu." That's because in some percentage of cases, the vaccine will not be effective because the virus has mutated. But it doesn't mean that the vaccine doesn't work. Okay? So bear that in mind. So how is it that the virus can mutate and escape from these antibodies that are blocking the interaction or blocking the virus entry? And it can, but however, the virus can mutate and escape from neutralizing antibodies, but it still remains infectious. So what we want to do here is look at the sites where the antibodies, neutralizing antibodies, bind, and where the receptor for influenza virus binds on the hemagglutinin protein. So this is the HA1 subunit of the hemagglutinin protein. So this is the globular head domain that binds to the receptor. Sialic acid binds into this pocket here, you see. And the residues that are mutated that allow escape from neutralizing antibodies are shown in their different colors on the surface here. So this explains why the virus can mutate these residues, can escape antibody binding by mutating these residues. But the pocket that is involved directly in binding sialic acid is not modified. So the virus retains the ability to infect cells even though it escapes from neutralizing antibodies. Now, that being the case, there are several approaches that have been attempted to try and look and find out whether we can get a universal influenza vaccine. That is to say, a vaccine that would be effective against different influenza strains, that would be effective against 100% of the viruses that are in circulation and would not need to be changed every year or two. Now, when you look at the structure of the influenza virus particle by electron cryotomography, this is the kind of thing that you can see. So the surface of the particle is really covered by the hemagglutinin in green and the neuraminidase molecules. And the head groups really cover the whole of the particle. So if there are any neutralizing epitopes that are down, right at the base of these hemagglutinin molecules, they can't really be accessed by antibodies. So the other neutralizing antibody epitopes may be hidden by the presence of hemagglutinin and neuraminidase. Now, the question is, are there other neutralizing epitopes on other virus or parts of the external region of the virus particle that are hidden by hemagglutinin and neuraminidase? And in fact, there are. One of them is the minor matrix protein M2. It's a proton channel. It's targeted by the antiviral drugs amantadine and rimantadine. If its function is inhibited, the virus cannot uncode correctly and infection is blocked at an early stage. Normally, the M2 protein is relatively poorly immunogenic, and people who are infected do not develop neutralizing antibodies against this protein. So here are the team of researchers who have tried to develop a way to make the M2 protein more immunogenic, and they did that by producing virus-like particles by expressing the M1 and M2 virus protein. So here if they do that, then you get particles of pretty much the same size as an influenza virus, but they don't have any hemagglutinin and neuraminidase on the outside. So the M2 protein is much more accessible, and people, or animals at least, who get vaccinated by these M1, M2 VLPs develop higher titers of antibodies against the M2 protein. Can this work? Can this give you a universal influenza vaccine? Because the M2 protein does not vary over time amongst influenza viruses, so it's a stable target. Here are a couple of experiments in mice where they vaccinated mice with standard vaccine strain of influenza virus, and they challenged the mouse with a highly pathogenic H5N1 N1 or with the new H1N1 influenza from 2009. So for the highly pathogenic viruses, okay, non-vaccinated mice, they all get very, very sick. If they have the standard vaccine, okay, they still get pretty sick and then recover. But if they have the standard vaccine together with the M2 VLPs, all of these mice were completely protected from infection. Now with the new H1N1 influenza, where you get a similar kind of thing, the mice which were not vaccinated got very, very ill. Standard seasonal influenza vaccine is very, very ineffective. Okay, this is this curve here. Mice are getting pretty sick. And protection was markedly enhanced by combining the influenza standard vaccine strain together with these M2 VLPs. So at least in this preclinical mouse model, this approach seems to be quite promising. The good thing about it is that you're not looking to replace seasonal influenza vaccine, but you're going to add something to it to make it more effective. Now, another kind of approach is to try and look for antibodies that bind somewhere on the hemagglutinin, but not on the head group. And this is from a review by Antonio Lancevecchia, published in 2013. So there was been considerable effort to try and define monoclonal antibodies against the hemagglutinin protein that have a broadly neutralizing activity. That is, they can neutralize many different strains of influenza virus. Now, when you look at where these broadly neutralizing antibodies bind, it appears that most of them seem to bind to the stem domain, which contains a stem region of the hemagglutinin, which contains the fusion domain. So this is HA1, this is HA2. So it's binding on the fusion domain, and it's going to prevent entry of the virus after it's binding to the surface of a susceptible cell. Now, most people do not develop high levels of antibodies to these regions of the influenza hemagglutinin. Probably because, this is the head group that's on the surface of the virus particle. It's more immunogenic. So the question is, how can you make this part of the hemagglutinin more immunogenic? This is two different approaches, okay? One of them is to try and change the immunogen. So this one was with naked DNA, uses a prime, and then boost with the seasonal influenza virus. So if you combine these two different forms of antigen, then it seems that you can get broad neutralization because this stem region becomes more immunogenic. So this is in phase 1 clinical trials at the moment. They're trying to see if this is actually going to work in human beings and will not just be effective in animals. Another way to do this was a very clever piece of work here, to try and combine the hemagglutinin extracellular domains to ferritin. Why did they do this? Because ferritin, 8 molecules of ferritin are going to combine into a kind of cubic structure. And in the cube, if you look at the vertices of a cube, there are like 3 edges that connect to each vertex. So this is a 3-fold axis of symmetry. That's what you need in order to form a trimer of hemagglutinin, which is the form that you find on the surface of a virus. So in fact, when they did this, they could form these kind of like strange looking structures here. That's the ferritin core, and each one of these small spikes here is a trimer of hemagglutinin. Now you can see that in this kind of structure, the stem domain is much more accessible than what you have on the surface of a real virus particle. And in fact, when they used these ferritin hemagglutinin nanoparticles as an immunogen, they got much, much higher neutralizing activity. And this is because they were able to induce the presence of stem-specific antibodies. So these different vaccine approaches towards vaccination may lead to the development of much more effective influenza vaccines in the future. However, one thing you should always bear in mind as clinicians for any kind of new medical intervention, whether it's a therapy or a preventive strategy, is my intervention going to be safe? And this is a question which will need to be posed for vaccines, just like for any other kind of medical intervention. And there are some experiments, in this one in pig, to show that perhaps we need to be a little bit careful. These were experiments that were done to see whether vaccination could protect pigs from the new variant H1N1 swine flu that's been in circulation since 2009. So when pigs were infected with this strain of influenza, They got sick, they got influenza. This was the gross anatomy of the lungs here. And by histology, you can see that there's quite a bit of inflammation around here. Look at all these, probably neutrophil, maybe some lymphocyte infiltration here. But you still have the alveolar structure, which is maintained. However, when they vaccinated before they infected the animals with the new variant influenza, then they found really much more severe pathology. Look at these regions of necrosis here in the lung. And histologically, you have a real big problem here. Okay. Now they were able to show that this enhanced pathology was due to the development of stem-specific antibodies, which were induced by vaccination. So this is one thing that will have to be studied carefully, is to find out whether new vaccine technologies are going to be safe. This is something we'll need to find out even before we ask the question of efficacy. Okay, phase 1 trials need to give an answer on this question. Okay, so that's pretty much where we are with influenza in terms of the problem of antigenic drift and what the current strategies are to try and develop more effective vaccines. Now in the HIV field, there is a similar kind of problem. Okay, so as you know, there is no effective vaccine available against HIV at the moment. And in fact, people who are infected can become super infected by a second strain of HIV. And that means that there's some kind of problem with the type of neutralizing antibodies that are developed in a HIV infected person. So this is a very nice set of experiments from Doug Richmond's lab. And they took plasma samples from the same person at different times after seroconversion, and they isolated virus from the same blood samples. And then they measured the neutralizing activity of the plasma against the viruses that were present at different time points. So at seroconversion, okay, or before seroconversion, right? This is a time 0 here. There are no neutralizing antibodies against any of the viruses, even though virus is present. 3 months after infection, you have neutralizing antibody against the virus that was present at the time of infection, but kind of lower neutralizing titer against the virus that's there at 3 months. At 6 months, you developed antibodies against the virus that was present 3 months after infection, but you've got no neutralizing antibodies against the virus that is actually circulating at this time. And so it goes on. If you look at the diagonal here, the virus is always 1 step ahead of the immune response. Okay, so 2 years after infection, you've developed a relatively pretty high neutralizing titer against all of the viruses that were circulating up to about 18 months after infection. But there's no neutralizing antibodies against the virus that is actually circulating in the patient at that time. So what does this tell us? This tells us that HIV-infected patients naturally develop neutralizing antibodies, but most of these neutralizing antibodies are strain-specific. They are not broadly neutralizing. And the virus is continuously mutating and escaping from the host immune response. So you have antigenic drift, again, this time inside an individual over the course of a chronic infection, not at the level of the population, but in each single person who is infected. However, some broadly neutralizing antibodies do exist against HIV glycoprotein. To understand how they work, we have to remind ourselves a little bit about the structure of the HIV envelope glycoproteins and what their role is in attachment and entry. So the ENV precursor glycoprotein is cleaved into GP120 and GP41, and the functional form of GP120 and GP41 is a trimer. So on the surface of the HIV virion, you have GP41 and GP120 trimers. GP120 is the binding subunit. So in the first place, it's going to bind to CD4. This binding induces liberation of the V3 loop, which binds to the co-receptor, which is CCR5. This step is necessary for the deployment of the fusion domains, which are in the GP41 trimer, which are responsible for membrane fusion between the virus envelope and the host cell plasma membrane. And that's what allows the virus capsid to enter into the infected cells. So there are 3 steps where neutralizing antibodies can block HIV entry, either by blocking the CD4 GP120 interaction, by blocking binding to CCR5, or by blocking membrane fusion by binding to GP41. If you look at this model of GP120, the GP120 and GP41 trimer, this shows you where the binding sites of different classes of neutralizing antibodies are. Now the V3 loop antibody is a neutralizing. They're going to bind to the V3 loop and prevent the interaction between the V3 loop and CCR5. However, V, right, here this means variable. This is a highly variable structure. So these neutralizing antibodies are strain specific. They're not broadly neutralizing. Broadly neutralizing antibodies mostly either bind to the CD4 binding site, so they just bind here and cover up this site, or they can bind to the stem region and they will inhibit fusion. So a lot of work has gone into finding the very rare antibodies that are broadly neutralizing, finding out where they bind. So now we know that these things exist. They can be formed under some circumstances. Can we induce these broadly neutralizing antibodies by vaccination? That's the question. Okay, so the biggest HIV vaccine trial that has ever been conducted is the RV144 trial. This was a phase 3 randomized clinical trial which involved 16,000 volunteers, more or less. 8,000 volunteers were injected with the vaccine. 8,000 volunteers got the placebo. Now, just a word about the design of this trial. Okay, these people who were volunteers for the trial, they did not get infected by HIV after they received the vaccine. They got counseling for methods of prevention of HIV transmission, and then they went back out into the countryside, out to the places where they lived, and they got on with their lives. Then they were monitored over time to find out how many seroconversions occurred in the vaccine group and how many occurred in the placebo group. And this trial was conducted in Thailand, I think over 5 years, from 2003 to 2008, maybe slightly wrong about that. Well, this trial was very, very encouraging because it was the only HIV vaccine trial where there was some significant protection in the group of people who had received vaccine. Okay, so this was partial, not 100% protection. And I think it's fair to say that we still don't know the mechanism that was involved here, because when we looked for, or when the investigators looked for broadly neutralizing antibodies, they were not found. So currently, none of the HIV candidate vaccines that have been tested in phase 2 or phase 3 clinical trials have been able to induce broadly neutralizing antibodies. Okay, so there are some strategies that are in preclinical development to induce broadly neutralizing antibodies by vaccination. So here's a consortium of different groups who are trying to redesign the GP120 trimer in order to make the CD4 binding site more immunogenic, so that broadly neutralizing antibodies against this site can be induced reproducibly by vaccination. But this is going to involve large-scale remodeling of the immunogen. As far as I am aware, there has been no clinical trial of the immunogens that they are producing, and the preclinical work has some kind of a problem because the germline V and J regions in the immunoglobulin locus in primates is not exactly the same as what you have in humans. So that makes it rather difficult to know whether, that is, even if you have success in a preclinical model, it's going to be very difficult to know whether that's going to be predictive of success in human beings. So another approach is to try and use virusomes in order to generate, so that means some kind of virus-like particle with peptide antigen, which is able to induce broadly neutralizing antibodies against the epitopes in GP41. So these are going to be binding to the stem region, inhibiting fusion. So there are encouraging results in preclinical trials in macaques, and I think this approach has been tested in phase 1. So safety and to some degree the immunogenicity has been confirmed for this kind of approach. Okay, so I'm going to finish up here with the summary of everything you need to try and remember about immune responses to viruses. So how does a cell know that it's infected? This depends to a large extent on the detection of viral nucleic acids by sensors, toll-like receptors or cytoplasmic sensors. Once this occurs, once viruses have been detected, there are 3 important consequences. Secretion of interferons, make cells more resistant to viral infection. Maturation of dendritic cells. This will help the adaptive immune response to be initiated. And thirdly, programmed cell death in the infected cell. This is important to try and limit viral replication and virus spread in the organism. Okay, for the innate responses, interferon and apoptosis. Interferon, never ever forget that interferon has no direct effect on the virus. It works because it can induce an antiviral state in the host cell. Still in clinical use for the therapy of hepatitis C and hepatitis B infections. Cellular immune responses, how does this work? Cytotoxic T lymphocytes and natural killer, that is NK cells, can recognize and kill virus-infected cells. They have an important role in limiting virus replication in acute and chronic infections. But it appears that in humans, at least, they are not sufficient for effective protection after vaccination. Finally, humoral response. Remember what neutralizing antibodies are, how they function. There are 2 kind of applications very important clinically. One of them is the use of hyperimmunoglobulins for post-exposure prophylaxis. And secondly, hyperimmune immunoglobulins or monoclonal antibodies for post-exposure prophylaxis. And basically the general idea that all the vaccines that we have available now that are effective work by inducing neutralizing antibodies. And once you've understood that, then that helps you to really understand why vaccines do not exist currently for some viruses. Okay, that's why we still don't have an effective vaccine for HIV infection. So I'll stop there, and I hope you enjoy the JPEMS, the rest of the JPEMS.